Biomolecule we're going to officially talk about are the carbohydrates. So carbohydrates are the main fuel source, and they're also a building material for the cell. So, mitochondria, powerhouse of the cell, like that glucose that we put into the mitochondria is an example of a carbohydrate. And remember, we're talking about monomers and polymers. Monomers make up the polymers. The monomer for the carbohydrates are the monosaccharides. Okay, I'll start kind of getting a list here. So our first biomolecule is the carbohydrate, and then the monomer there, is the monosaccharide. So we'll kind of keep adding to this list throughout the unit. And then the polymer, remember poly means many, that is the polysaccharide. So just some basic names you gotta kind of memorize it. It's freshman bio review. That is something your freshman bio teacher should have had you learn. And the name kind of gives it away, right? Mono means one. Saccharide is referring to the sugar. So poly, many sugars. Okay. Cursor over here. So let's look at some examples. The classic, because one of the things you have to be able to do, as you learn the other three biomolecules, I show you a structure like this. You've got to be able to say, hey, that is a sugar. Now, you don't need to be able to say that is a triose sugar, but just in the spirit of like thinking, starting to begin to think in a kind of organic chemistry way, a triose sugar, tri means three, think of a triangle. It is a 3-carbon sugar. And then notice the functional group that they highlighted there. Anybody remember what functional group that is? Yeah. No, close. It's part of it. Rebecca? No, that's what she was saying. That is part of it. That's what makes the carboxylic acid one tricky, is because it has this, but it also has an OH at the end there. Yeah. Carbonyl. Carbonyl, yeah. Mm-hmm. Good. Oh, is that? Oh, I'm sorry. That's what you said. Okay, my bad. I thought you said carboxylic. Carbonyl. What? Then there's two kinds of carbonyls. Which one would this be? Think of like back. Yeah. Aldehyde. Yeah. Remember, ketone would be if it was like that same structure, but like in the middle of it. So, okay. Anyways, this is specifically glyceraldehyde. Don't even know that name. Just an example of one of these sugars. And it's part of the cellular respiration process where you're breaking down that glucose. It's one of the intermediate forms. Ribose, like in a ribonucleic acid, this is the ribose sugar that makes up the RNA nucleotides. And it's a pentose sugar, so 5-carbon. Sugars, you're always going to see, and this is really important, sugars always have, let me write it more up here. Think CHO, like so it's carbon, hydrogen, oxygen, but it's specifically like a 1 to 2 to 1 ratio. Meaning, like if you look here, you have 1 carbon. For every 1 carbon and 1 oxygen, you have 2 hydrogens. And at the pentose, like you have twice the number of hydrogens as you do carbons and oxygens. Okay, simple enough. And then, yeah, they have this ketone group. Oh, what other functional group do you see here? There's 2 functional groups that are really key with the carbohydrates. Yep. Yeah, the hydroxyl group, this OH. Also called the alcohol. Okay, cool. Anyways, another example would be a hexose sugar. So, like glucose, fructose. Maybe you've heard of fructose before. It's a kind of a sweet. I mean, all sugars are mostly sweet, but like it's just a kind of fructose will be used in a lot of our food products and stuff. Anyways, these ones are all 6-carbon. Again, notice that the 1 to 2 to 1 ratio, and then fructose is interesting because that has like an actual ketone in the middle there. Okay, so let me make sure I said everything here. Monosaccharides. Yeah, I guess a simpler way is just this. 1 to 2 to 1. Glucose, the most common one. And yeah, that carbonyl group. Okay. Now, what's interesting with the sugars is how I just showed you those sugars was the linear form. So sugars have a linear and a circular form. What I mean by that, if we take this linear, I believe this is glucose. Yep, glucose. This glucose, when you put it in an aqueous environment, what's an aqueous environment?
Water.
Water, yeah. You know what I told my chemistry students? Like for years, I was like, aqueous. You can remember it's water because Spanish for water is agua. And what's the problem with me saying that? Agua is spelled A-G-U-A, not A-Q-U-A. So anyways, whatever. Okay, aqueous is in water. So you put this linear sugar in water, and it's going to fold up into this ring structure, where the number one carbon, so you number from top down, the number one carbon, this aldehyde group is going to bend in and connect with this alcohol, this hydroxyl group, and then you then get a covalent bond that's going to form right there. Okay, so there you go. You don't need to memorize like that it's the one going with number 5 carbon. If you do biochemistry, like your junior, senior year of college, you do bio, they'll make you memorize these structures, draw the ring and the linear but like I just kind of want you now to start kind of thinking through what this would look like. What do these double arrows mean?
It's a reversible reaction.
Reversible, yep. Now what's interesting is like they say in an aqueous environment it favors the ring structure. What's the problem with like how this slide has these arrows if it favors this ring structure?
It goes both ways.
It does go both ways, but if one way is favored, how do they represent that in chemistry? Or how should they? It is in equilibrium, but like something with the arrows, yeah?
It would just be one arrow to that way.
Not just, but you're not just one arrow, but I think you're thinking along the right lines. One arrow is bigger. So like this arrow should be like that. And then the backwards arrow is still there, but it's smaller, saying some of these unfold back to the linear structure, but not as many. So anyways, not that big of a deal, but I just thought it was worth pointing out. Okay. Now, if I go back real quick, here they labeled all of the atoms, right? But we talked about last class that, we can all be adults here, and to save time, we don't have to literally label all of the carbons. We can assume that these edges, or these vertici, those are carbons. So there, that's a carbon, that's a carbon, that's a carbon. Now here, because this corner there actually isn't a carbon, it's an oxygen, they will then label it something else. Okay? I also told you a lot of times they will leave off the hydrogens. You could leave off the hydrogens here, but they usually don't because they want to show those hydroxyl groups. But anyways, this is the abbreviated structure of it. Okay, let me make sure I set everything. That's good. Okay, so now we're going to talk about how do these monosaccharides, so like glucose and fructose, those are monosaccharides, how would they then form polysaccharides? And they're going to do that through the dehydration synthesis that we went over last class, right? So remember, you're going to remove a water, and then they're going to form what's called a glycosidic linkage. So you need to know that word. So for all of these biomolecules, you've got to know their covalent linkages, meaning like how the monomer becomes a polymer. You need to know the specific names of them. So for, for carbohydrates, it's the glycosidic linkage. And then where the glycosidic linkage occurs at, they'll name it based on the carbons that they occur at. So for example here, the glycosidic linkage is between the number 1 carbon and the number 2 carbon of the 2 monosaccharides. And then they're linked here with this little oxygen here. Okay. There you go. If you're kind of curious, like you'll notice that the placement of these hydroxyl groups, like they can go up and down. That can be a big difference in the actual structure of one monosaccharide versus the other. Is like the ordering of these hydroxyl groups going up versus down. If you do biochemistry, they'll make you memorize the structure of glucose, structure of fructose. You have to actually draw these reactions. for us, you don't have to draw them. I just want you to know the name of it and understand that if I say 1 to 2, you know what, like you can kind of picture what that is saying from the ring structure. But you don't have to literally draw it. Just pick it out amongst the other ones. Good? Okay, let me make sure I said everything. Oh, if there's 2 of them, that's a disaccharide. Make some common sense. Di means 2. So. Yep.
Wait, what you mean? Like, so, sorry, when you go back, the 2 of them are bonded, so is that a disaccharide?
This would be a disaccharide. So sucrose is an example of a disaccharide.
Okay.
Yep. But glucose and fructose, those are monosaccharides.
Oh, so it's the 2 of the same.
These don't have to be the same to be a disaccharide. You could have 2 different monomers, 2 different monosaccharides like glucose and fructose will come together to make sucrose. I don't know if that's your question.
Okay.
Meaning, like, it doesn't have to literally be two glucoses coming together to make a disaccharide. A disaccharide could be 2 fructoses, 2 glucoses, 1 glucose, 1 fructose. All the slide where I'll show you the names of some other disaccharides that are named based on the monosaccharides that make them up. Yep.
So my question kind of relates to the last session too, but
Yeah.
why is this event called a dehydration synthesis? It
Oh,
shouldn't
sure.
technically be decomposition or like neutralization because it's like takes out water.
Oh, I see what you're saying with neutralization. The dehydration is coming from the fact, like, if you're dehydrated, you're you're losing water. So these molecules are losing a water. So that's where the name's coming from. This wouldn't be an acid-base reaction, so that's why it would be neutralization. Like, there's not really like a true like giving or receiving of like the hydrogen ions. But either way, dehydration. That's why they call it. And synthesis means you're building something, and then dehydration, you're removing the water there. Does that help? Yeah. Sometimes at some point somebody just chooses the name, they just kind of roll with it. You know. Are we good? Other questions? Okay. My clicker will. All right, let me make sure. Oh, the ose ending. So ose means like carbohydrates. So they always have that ose ending. And then again, make sure you know the names of those covalent bonds for all the other biomolecules, which we haven't talked about yet, but we will. Okay. So as I was saying, there's a bunch of different other like disaccharides. So we talked about sucrose, which is a combination of glucose and fructose. There's also called lactose, which you probably have heard about, right? Like lactose-free milk. So in lactose, lactose is the sugar that's in milk and other dairy products. That's made of the monosaccharide galactose, which is pictured here, and glucose, which is pictured there. So yeah, people who are lactose intolerant, they basically have an issue breaking apart that glycosidic linkage. The lactase is the enzyme that helps break this apart. And so just the structure of that enzyme, this just doesn't fit in there either at all or as efficiently. And so that's why there's different degrees of lactose intolerance. Some people, their enzymes work kind of well, but not as well as like how they should, so they're slightly intolerant. Others, their lactase enzyme is either not made at all, or again, it's like just not formed properly. So this literally just will not fit. You know, like just as like mechanically as like this cap won't fit on the marker anymore. So, and then maltose, that's glucose plus glucose. As I'm saying here, I don't need you to memorize these actual structures. I just want you to know like sucrose, lactose, and maltose. Those are disaccharides. They're not polysaccharides or monosaccharides. But you don't need to know that lactose is galactose plus glucose. Just that lactose is the disaccharide. Okay. In other words, I want you to know the 3 monosaccharides are glucose, fructose, and galactose. Okay. And then you combine these together, and you can make these different disaccharides. Okay. And it's worth pointing out, like especially looking at glucose and galactose, their structures are very, very similar, like almost identical. But like notice how the hydroxyl groups in glucose at the 1 and the 4 position are pointed down, whereas for galactose at the 1 and 4 position, the carbon number, they're pointed up. So just small differences like that can make a big difference in how that molecule behaves, is the point there. Is it good? I'll show you the polysaccharide examples in the coming slides, but just for now, these are the monosaccharides and disaccharides. It's not like an important detail necessarily for like the test. So I was just saying like people who are lactose intolerant, they can't break apart that glycosidic linkage. Just how it's positioned, that this will have to like fit into like enzymes, these proteins, like a three-dimensional shape, and just the shape of some people's enzymes is just doesn't work properly. So this won't fit on there. Kind of like the cap won't fit on this marker, so it just keeps falling off. It's kind of like that. Yeah. Okay. All right, now the polysaccharides. So polysaccharides would be, you move beyond disaccharides. So basically polysaccharide they define as like 3 or more monosaccharides. You know, because they're not going to like say like trisaccharide, quad saccharide. Eventually they say, all right, it's just a polysaccharide. There's just many of them. And the polysaccharides store their function is way more in storage and then structure. So storage and structure is what you want to think of when you think of polysaccharides. Okay, not so much energy. The monosaccharides and disaccharides, those have more of an energy role, especially the monosaccharides. But the polysaccharides are like, hey, we ate a ton of pasta. This is the idea of like, you'll hear like runners, they'll do like the pasta party. You ever heard that? Where they're going to like carb load. The idea is, hey, we eat a ton of pasta. We're going to store all this these sugars up as these polysaccharides, and I'll show you like the different kinds of them. And then as you're running, your body starts breaking apart those polysaccharides, and they give you energy. I don't know how much that sounds good in theory. I don't know how much that actually makes a big difference, but you know, that's the idea. Anyways, but I talked about the glycosidic linkages. So as you'll see in the next slide, as the structure of these polysaccharides, I'll just jump there now. Where you place these glycosidic linkages, that's going to be the big difference in how you actually make the polysaccharide. Meaning the difference isn't in like the monosaccharide that makes it the polysaccharide, it's how do you link the monosaccharides. So for example, in potatoes. Potatoes have tons of glucose that's just stored up for that plant. That way if the plant goes through a time where it can't, you know, there's no sunlight, the plant can't do photosynthesis, it has a bunch of reserved carbohydrates there that the plant can use to get energy. And so you'll basically take all these glucose monomers and they do the glycosidic linkage, there's your glycosidic linkage, and they just keep combining those together and together and together, and then that is what we call starch. So you probably have heard the word starch just in everyday language. All starch is just a bunch of glucoses linked together. So it's nothing like, there isn't like some unique like molecule necessarily called starch, it's just a bunch of glucoses together. Now, as is glycogen. So our plant's version of stored carbohydrates is starch. Our version of it is glycogen. So same thing. It's still glucose. It's just when it's in our muscle cells, we call it glycogen. Now, structurally, what is a little bit different is that we do more branching. So for muscle cells, it's heavily branched, and that begs the question: why do these polysaccharides want to branch in the first place? What's the idea of branching? Why not just make them like, in other words, why not make them just giant chains? Why do they bother like branching them apart? What would be the idea behind that? Yeah.
Maybe because they intertwine.
Yeah, exactly. Yeah. Yeah, it's like an efficiency thing, right? You can fit more in here if you can have these kind of branch structures where they can then kind of link together. The biggest reason would be efficiency in breaking it down. Because if it was one long chain, you could only break that chain at one spot. Whereas the more it's branched, the more entry points you have to break this apart. Think of like a road. There's only one entrance ramp to a highway. It's going to get plugged up a bit, but if you have multiple entrances and exits, it's going to be a little more efficient for entering and exiting the highway. So that's the idea of the branching, is especially the efficiency and trying to break it down. So, and then why is the glycogen stored in muscle cells for humans? Why would you want to store it in the muscles? Remember, the glycogen is stored glucose, which is your main energy source. Like when they say mitochondria is the powerhouse of the cell, where's the power coming from? It's coming from glucose. So why would you want to store the glucose in the muscle cells as glycogen? Why not store them in the fat cells or the skin cells. Yeah.
Because they won't need to travel as far. Like the energy doesn't need to be, brought from one place to another to the muscle in order for the muscle to use up energy.
Yeah. I think what you're leaving out there is like you need, like if your life's in danger and you got to run from a tiger or like something else, your muscles are taking the biggest priority there. So you want to make sure you can get that energy supply right to your muscles, and not, as you're saying, have to like transport it from like, your skin, where it's like how much you're using glucose there, but not as much as like the muscles are using. It wouldn't be as efficient to store it there, right? It would be like me storing the world history books in my room. It's like you could do that. I have like just cabinets in here, but why would you do that? Like, why not keep them in the world history classes? It's kind of maybe the idea, or maybe like the library is a better example. I don't know. Hopefully you see what I'm saying. But anyway, starch is the storage form in plants. Glycogen is the storage form of glucose in animals. So make sure you know those differences. Okay, questions here? Don't worry about amylose versus amylopectin. It's not, for our purposes, not that important. It's just the difference in like just the extent of branching. If you get into plant biology, they'll worry way more about it, but AP exam, they're not really getting to that level of detail. All right, let me make sure I said everything here. Yeah, and that's all that starch will be stored in these things called granules. That's what I was saying at the beginning of class with the bananas. So that starch will be broken down in different ways depending on the ripeness of the banana. So that's what we're going to learn about. You're going to get to like kind of study and look at next class. But yeah, so then as the animal or the plant needs that stored sugar, it'll then have enzymes that hydrolyze. Remember hydrolysis? That's how you break apart the polymer to the monomer. So as the plant or the animal needs that carbohydrate, it'll hydrolyze the polymer into the monomer glucose for it to use. Yep, said everything here. Glycogen is the form for animals. Liver cells also have a lot of glycogen. Why would that make sense? What's our liver important doing? Yeah, your liver does a lot of breakdown stuff. So toxic things or just basic breakdown products of your food goes through your liver. So your liver uses a lot of energy. So that's why you're going to want to store a lot of the glycogen there. Okay, now then there are structural polysaccharides. So we talked about storage polysaccharides. This would be an example of a structural polysaccharide. Now, we as humans, we don't really have structural, I mean, we technically do, but it's not as common as plants because we have a skeleton. Remember, plants don't have a backbone. So they have what? What's the difference between a plant cell and an animal cell? Basic cell wall, yeah? So you want to look for the cell wall to see a plant cell from the animal cell. The cell wall of the animal cell is made of cellulose. So the cellulose is all these glucose molecules that are linked together in the polysaccharide. But now what you don't see is there is no branching. They are unbranched. And these single chains are going to line up with each other, and they form hydrogen bonds to strengthen the connection between those chains. Now, a big concept in all of biology is structure fits the function. If something has a certain structure, it's because it has a certain function. So how does the structure meet the function here? Why would cellulose have these unbranched chains rather than the branched chains that the storage polysaccharides had? Think about that.
That helps to keep the planet more rigid.
How would the unbranched do that versus like a branched one? You're right. Yeah, this would be stronger. Yeah, because like they're literally like you're maximizing the contact between the strands by making them just chains. If that makes sense. Whereas if they're branched, there's going to be many parts where like those branches don't actually connect with each other. Think of like if you have a big old stack of like brush, like twigs and stuff that like you're trimming trees and stuff. The stack, like you could step on the stack and squish it down. That's because the branches don't all connect all together. Whereas if you were to take those branches and maybe you see people like tie them together with twine, that's kind of like what the plant does when they're making their cell wall. They're taking these unbranched polysaccharides because it's easier to tie them together, and then they can start forming the hydrogen bonding, and it's going to be stronger. is the point. Okay. Make sure I set everything here. All right. More of kind of like a fun fact kind of thing. Not something I really test much on, but there is actually 2 different forms of glucose. Now, I hinted at it earlier with where these hydroxyl groups are at. So there's alpha and beta glucose. Alpha glucose will point downwards. This hydroxyl group on the 1 carbon points down. Beta glucose, the hydroxyl group points up. And then technically, glucose can shift back and forth between the 2 forms. Now, what's going on with that is you can make, like the difference between like when you're making starch and cellulose, if you want to make starch, the storage polysaccharide, versus you want to make cellulose, the structural polysaccharide, you're using 2 different forms of the glucose monomers, alpha or beta. Don't need to memorize the difference, but starch has the alpha glucose monomers, so you have that 1,4-glycosidic linkage, whereas cellulose has the beta glucose monomers. And it's like, okay, cool. What we're building towards is this idea of fiber. So you've maybe looked at a package, food package, you see like fiber on the back of it. Or a mouse. Mice can like eat wood and stuff, and you're like, hey, why can't I eat wood? You know, that sounds pretty nice. Like, the reason why you can't eat wood is because you don't have enzymes that can break apart the 1 to 4 linkage, glycosidic linkage, between the beta glucose monomers. So basically, because these hydroxyl groups are located like this, our enzymes can't, it doesn't fit. Again, back to like the cap on like the marker, it literally just, the enzymes cannot wrap around this and break it down. But mice have the enzyme where they can break down the cellulose, and that's why they can just eat like a tree, and they can still like get the energy out of it. So there you go. So then that's the idea of fibers. It helps push waste through your body, right? You know, it doesn't break down all the way. That's because like the fiber of plants is made of the cell wall of the plants. That's why if you want to get more fiber in your diet, you're going to want to eat a lot of plants, because the cell wall in those plants is going to be that insoluble fiber, the fiber that can't be broken down all the way. Cool. Yeah, make sure I set everything here. Yeah, starch molecules are also going to have this helical kind of structure. But the important thing is really that cellulose has that straight unbranched chain. That way they can line up and package together really tightly. Got the hydrogen bonding between the parallel celluloses. As I said, our enzymes, we can break the alpha linkages, right, between like the starches, but we can't do the beta linkages between the cellulose, the cell walls of the plants. There are some bacteria that can digest cellulose, and then like when I say like mice or cows or termites, what's going on is they have basically the bacteria in their gut that can break down that cellulose. Okay. Symbiotic relationship. What's symbiotic relationship? What does that mean? Yep.
Like a relationship in which like 2 organisms benefit from each other?
That specifically is mutualism, which is a type of symbiotic. So what you said, like the first sense is just a relationship between 2 different organisms, 2 different species. That's a symbiotic relationship. And then there's like mutualism, both benefit what you said. And then you have like parasitism, one benefits, one doesn't care. Commensalism, one benefits, the other is just unaffected. Fungi, they have the enzymes able to break down cellulose. That's why fungi can help us recycle. And they're like the decomposers and all that. Okay, almost done, but one other structural polysaccharide that's in your arthropods is chitin, not chitin. Okay, now you're all thinking it. Chitin. Chitin is basically, it's kind of a similar idea somewhat to cellulose, but basically they can just form much more advanced chains. I found this picture online just if you wanted to just see it in a little more detail. All I really need you to know is that chitin is in, and fungi, But like fungi and then your insects, your arthropods, they have chitin, just like a different kind of structural polysaccharide. That's all you need to know about it. Don't need to know like any of the detailed structural information of it. Even though I don't know, it sounds kind of interesting. Learn more about it, but okay. So that'll do it for carbohydrates. So then I'll give you, and we'll go like 15, 20 minutes. We'll go 15 minutes. Work on the mastering bio, and then we'll come back and do the standard deviation.